Please confirm me if you are all again present, then we start. All is good. Let everyone join it again because we are about to start with PCR. We were done with the first tool, that is a gel electrophoresis. We might do them in detail soon. But there are 7 members. I don't know how many members were there before. So in total 13 students have joined this thing. So the best part is that this will be recorded forever in that molecular biology workshop. I have created one playlist, so you can join it anytime, watch it from there. Okay, so I think everyone has joined it again. So I continue. So I want to say about PCR. So the PCR is basically very versatile component. Mullis have also got the Nobel Prize winner for the finding it, and this allows a single DNA sequence to be copied or altered in the predetermined ways. And PCR has many variations like reverse transcription, RT-PCR or amplification of RNA, real-time PCRs, which allow for the quantitative measurement of DNA and RNA molecules. So basically in this cycle you have a denaturation, annealing, and elongation is happening at 3 different temperature like 90, 50 to 70 and then 70 degrees Celsius. So by there are copies are being produced of same DNA template. So 2 raised to power n in exponential way. So in the 2 copies, so 2 raised to power 2, 4 copies will produce, then 8, 16 and 32. So by the 35th cycle you have a 68 billion copies. By the 36th, for 35th cycle, we will have around 68 billion copies. So let's see more about that. So PCR, as we have discussed, short history, it was discovered by Carey Mullis in 1983. So the first publication was done of PCR by Cetus Corporation, which appears in Science. Then purified Taq polymerase is first used in the PCR. Then Perkin-Elmer introduces the automated thermal cycler. And then 1989, Science declares Taq polymerase molecule of the year. And the purpose is to amplify a lot of double-stranded DNA molecules with the same size and sequence by enzymatic method and cycling condition. So temperature that is required is for 92 to 94 degrees Celsius, and double-stranded DNA melts to make a single-stranded DNA. So from 92 degrees Celsius, it's make your single-stranded DNA out of it. Then annealing, we make a temperature of 50 to 70 degrees Celsius, depending on the melting temperature of the expected duplex. So primers bind to their complementary sequences. So it will have reverse primer and forward primer. Then in extension, temperatures of around 72 degrees Celsius, time is around 0.5 to 3 minutes, and DNA polymerase binds to the annealed primers and extend DNA at the 3' end of the chain. So you have your D nucleotide pairs here, and the Taq polymerase being attached there. So then products of extension. So afterwards you will have your DNA completed produced by this, and then as we have discussed, by the end of PCR you will have millions of copies. In the chemical components, it includes your magnesium chloride, buffer, DNTPs, primers, DNA polymerase and target DNA. So all are required, and the basic requirement for PCR reaction is DNA sequence of target region must be known, and the primers, that is typically 20 to 30 bases in size, and these can be readily produced by commercial companies. A thermostable DNA polymerase, that is Taq polymerase, which is not inactivated by heating to temperature 95 degrees Celsius, and DNA thermal cycler, machine which can be programmed to carry out heating and cooling of samples over the number of cycles. Then applications of PCR, the molecular identifications, sequencing, genetic engineering. So it has wide range of applications. So these are the instrumentations that we mostly use, thermal cyclers. So on the left one is the most common one that we use in the lab. This one is the least common, but this one has 4 copies of DNA, or 4 copies of thermal cycler in one machine, and this is the most latest one, light cycler being used. So just want to give you touch, and the primer that you want to use is nucleic acid that serve as a starting point for DNA synthesis, and these primers are usually short, chemically synthesized oligonucleotides with a length of about 20 base pairs. And the replication starts at the 3' end and the copies of the opposite strand. So yeah, we have discussed this. So let's watch so you will have the other view also and this one also as a presentation, PCR. Okay, so I take you PCR protocol part 1. So we'll go through a virtual lab now to understand it. So it's self-explanatory, just read it. Just a second, student. So far, since morning we had started, we had discussed about the genetic code, how it is produced. Then we have discussed about genome, what is genome is made up of, why is it so important, how we can characterize it, and then we have started our lectures with the tools in the molecular biology. So I have just discussed with you since morning about that how we can separate your DNA, your RNA, your protein based on their molecular weight and electric charge via gel electrophoresis. So we will know about, so for the in the case of protein, you can go for 2D gel electrophoresis. For the case of DNA, you can go for agarose gel electrophoresis. And just now I have discussed you about PCR, that is polymerase chain reaction. The basic principle behind it, that is your denaturation, annealing, and elongation. At the denaturation, we just had a temperature of 70 to 90 degrees Celsius, in which we separate your concerned DNA template, and then at 50 to 72, annealing is done. Your forward and reverse primaries got attached. Then at the elongation, your Taq polymerase will come, and with the help of various DNTPs, there is trinucleotide phosphates. So DNA dinucleotide triphosphates, DNTPs, it will start producing the corresponding sequence. So your PCR, you will have by the 35th cycle, you will have in exponential way around 68 billion copies of your DNA. So that's the basic function of your DNA and of your PCR, sorry, and it would have many applications from diagnosis, from site-directed mutagenesis in medical field, and so on. So that's we had discussed so far this morning. Now I want to take you to the virtual lab of PCR, how PCR is done in the lab. So the lady will explain here very nicely in practically when you have got a chance to do this PCR in the lab. First of all, you need to manage. Let me start now.
Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a multi-sample procedure to avoid excess pipetting and potential errors. In this example of Protocol 2A, 4 students are amplifying the actinin 3 gene from all 4 of their DNA samples. This information is filled out on the left-hand side of the worksheet. On the right-hand side, students use the equations to determine the number of tubes needed and the multiplier for reagent volumes. In addition to the 4 DNA samples, a negative control sample is always included. Therefore, 5 tubes are needed for the PCR reaction. In the creation of the cocktail, pipetting error is accounted for by adding another sample. Therefore, the multiplier for this group is 6. Each row of Table 1 represents a common reagent used in all the samples undergoing a PCR reaction. The multiplier is filled out in all rows of the multiplier column of Table 1. Moving across each row, the volume indicated per reaction is multiplied by the multiplier. .5 times 6 equals 75 microliters of red tack added to the cocktail. 11.5 times 6 equals 69 microliters of water added to the cocktail. 0.5 times 6 equals 3 microliters of each of the forward and reverse primer added to the cocktail. Finally, the sum of these volumes should be equal to the product of the final row. 75 plus 69 plus 3 plus 3 equals 150 microliters of total volume in the cocktail. This amount matches 25 times 6. Table 2 is used as a guide for labeling of the PCR samples. As indicated by the equations, this group will be using 5 tubes. The class must collectively number the PCR samples to not confuse samples amongst the groups. In this example, these students have been assigned samples numbered 5 through 9. The sample names are filled out in the second column, and the primers used for each sample are filled out in the last 2 columns. In this example, the forward primer is actinin 3F, and the reverse primer is actinin 3R. The same primers are used for all samples. The common reagents provided are molecular biology water, a forward primer, which you should be sure to check for the marking of PCR to ensure that you are using the appropriate concentration. A reverse primer, and red tack ready mix. The red tack will need to be thawed prior to use, but once thawed, keep the solution on ice to prevent enzyme degradation. Next, prepare the cocktail by using the volumes and reagents indicated on the worksheet. Obtain a new tube and label it as the cocktail. When creating a cocktail, it is best to start with the largest volumes. Though red tack is the largest volume, the solution should be kept on ice as long as possible, so instead the water is added first. When adding each of the reagents, be sure to use the appropriate micropipetter for the volume being added. Next, the primers are added. It is best to pipette small volumes directly into larger volumes to avoid loss of the reagent. Finally, add the red tack and immediately return the stock to ice. Mix the solution and centrifuge all cocktail tubes created by all groups in the class together. Press and hold the short button to do a brief spin. The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of tubes. In this case, to amplify actinin-3, 24 microliters of cocktail solution is added to each of the 5 tubes. Before adding DNA to the PCR reactions, make sure that the tubes are labeled. Make sure all DNA samples are mixed before transferring the solution. One microliter is a very small volume, but you should still be able to see a drop in the tip. Put the tip all the way into the bottom of the tube and pump up and down on the plunger before expelling to the second stop. This helps to ensure that the 1 microliter of DNA has been expelled into the reaction. Cap the tubes tightly and flick to mix the samples. Use the mini microcentrifuge with the 8-tube strip head to collect the solution at the bottom of the tube. It is good practice to always mix and collect solutions before every step, especially if the solutions have been sitting for a while. Turn the thermocycler on with the switch in the back. Place the tubes in the thermocycler by pressing the metal latch and lifting up on the lid. Tubes can be placed in any spot or orientation. Close the lid and make sure the metal latch has clicked into place. Heat cycling programs have already been programmed into the thermocycler, so all you need to do is run them by pressing the Proceed button. Use the horizontal arrows to navigate to the program of interest, in this case ACTN, to amplify the actinin-3 locus, and hit Proceed. The heated lid should be enabled by hitting proceed to prevent extreme condensation at the top of the tube. The thermocycler will ramp to the initial temperature and begin timing the incubation. It is good practice to make sure the machine reaches this step with no errors. After it has initiated properly, the machine can be left while the reaction proceeds for several hours. Once the run is complete, the cycler can be turned off by simply flipping the switch in the back of the machine.
So students, there is some, yeah, it's happened universally. The internet start to give results very late. So what I'm speaking, I think it's not going through to you. So there's a 5 minutes lag here, I can see. Right? Can you, if I'm saying here, Give me a thumbs up at the my time is here 11:58. I want to check when did you reply me. So reply me with a thumbs up in return so that I can see how much lagging is there. Shall I continue this class or should I make a new connection? Because our topics are going done very nicely, that's no problem. I want to take you to the markers now. Okay, my signals I think are going really bad. Now yeah.